brought to you by LIPID MAPS.
Welcome to the LIPID MAPS Podcast. I'm Matthew Conroy, and today we're joined by Corinne Spickett, who is in the School of Biosciences and the Aston Centre for Membrane Excellence at Aston University here in the UK. Welcome, Corinne, to the podcast.
Oh, thank you, Matt. Thank you for inviting me.
Your research focuses a lot on redox biology. Could you give us a quick overview of what that is and how lipids relate to redox biology?
Well, redox biology is all about the balance of redox, which is oxidation and reduction in the cell. But it's not so much about bioenergetic redox and metabolism. It really focuses a bit more on antioxidants and their balance with oxidants that are produced adventitiously in cells. And the oxidants that are produced, if they're not detoxified or removed by antioxidants, can then lead to oxidative damage and oxidative stress. And the damage can be to all sorts of macromolecules like proteins and DNA, but also lipids and phospholipids. So that's where the lipids come into the process.
So it's vitally important to remove these species in a controlled way.
Yes, that's the basic idea.
And a lot of your work involves protein lipoxidation and structure-function consequences of that process, oxidative modifications of proteins. How are proteins modified by lipids? And could you give us a particular insight from your work?
Okay, so that's quite a complex story. So maybe I should say a little bit more about lipid oxidation first of all. So there are different ways in which lipids can be oxidized. I mean, there are many enzymes that oxidize lipids specifically, and they produce important signaling molecules for the immune system. But my focus was more on adventitious oxidation, so chemical oxidation that didn't involve enzymes. And that can be through oxidants that are radicals. And so the radical process of lipid oxidation initially generates hydroperoxides, and they can then be broken down or rearranged in many ways. You get a plethora of different compounds with different numbers of oxygens, and then the oxygens can cyclize and form epoxides and hydroxides if the hydroperoxide has been reduced, and also aldehydes. One of the things that can happen is that where there's a hydroperoxide, it can essentially break the chain of the fatty acid, and that yields aldehydes. There are some quite famous products of lipid peroxidation, one of which is 4-hydroxynonenal, which, as well as an aldehyde that's formed where it breaks down from the fatty acyl chain, it also has what's called an alpha-beta unsaturated group. So aldehydes are reactive compounds. They react with amine groups, NH2. So many proteins have amine groups on them, so lysines and arginines and so on. And then also the alpha-beta unsaturated group is reactive as well. That can react with amines as the aldehydes can, but it can also react with, as well as primary amines, secondary amines, such as in histidine, and thiol groups, such as cysteine. So you can then form these adducts between the lipid breakdown products with either the aldehyde or the alpha-beta unsaturated group, and they can form adducts on proteins. Some of them are stable, but they're not always that stable. So the aldehyde-amine reaction forms a Schiff's base, and they're quite readily reversible. It can be hydrolyzed. The Michael adduct is slightly less reversible, but can still be removed. So that's how you get what we call lipoxidation.
And is this usually bad news for the protein?
Well, that's a very interesting question. I think when we started working on this, we assumed that it was mostly going to affect the protein in a deleterious way and inhibit its activity. But actually, one of the things we've seen is that lipoxidation, and this is not just from my work, this is from lots of different groups, you can get many different effects. So, for example, it can cause the protein to unfold. slightly, or, especially if you've got a large bulky group that's got into it. And once proteins start to unfold, then their hydrophobic interiors tend to be more available, and then you get aggregation and precipitation, and it makes a bit of a mess. But on the other hand, some of the smaller compounds can get into the active site and interact with important catalytic groups such as cysteines in the active site. So then you tend to get inactivation, especially if it's an enzyme. So that depends on the enzyme, but usually it's undesirable. There are some cases where lipoxidation can increase the activity of the protein. And
That's surprising.
yes, and that's often in signaling pathways. So again, it's not from my work, but from a close colleague, Dolores Perez Sala, who's in Madrid. And she showed that HRAS activities in the RAS-MECA pathway was upregulated by adding lipid-derived compounds that contain cyclopentanone groups that are reactive. And the concept there is that by modifying the protein, you're actually directing it to a membrane site where it's more active. So it's not that it's directly activating the protein, but it's controlling where it goes to. And that's actually something that I think is still really interesting, how these oxidized lipid products affect how proteins are trafficked within the cell, whether they associate with membranes, which membranes, and there are lots of things we still don't know about this.
And I think that's a very important aspect of metabolism generally. It's not just how active the enzyme is, it's where it is and what
Yes.
it's doing. You might find an enzyme that is immensely active on a particular substrate, be it lipid or not.
Mm-hmm.
But if it's in a part of the body or a part of the cell that never sees that molecule, it doesn't matter how active it is.
Yeah, that's one of the things I remember discovering a few years ago, that so many proteins actually have what they call moonlighting functions. So glyceraldehyde 3-phosphate dehydrogenase is a glycolytic enzyme, which any biochemist learned about as an undergraduate.
Yeah.
And then you discover that A, it can be oxidized, and B, it can go off the nucleus and have a sort of transcription factor activity there, which just at the time seemed really amazing. But now we see that with a number of different proteins where you wouldn't expect them really to do that, and then they have all these pleiotropic effects.
So, I mean, that alludes to my next question. How has the field evolved over the course of your career? And where do you think it's going? What are the exciting frontiers we have in the next coming years?
I guess it depends which field are you asking about. Are you asking about the redox biology field or the lipidomics field, or shall I say something about each?
Or either of them, or the technical, I'm going to throw another issue in here, even the technicalities of mass spec.
Yeah, well, that's what I was going to say about lipidomics. When I started doing mass spectrometry and trying to analyze phospholipid oxidation, we had a single quadrupole machine, and we managed to measure some phospholipid hydroperoxides. And then since then, we went to multi-component mass spectrometers with triple quads and quad TOFs, and then Orbitraps were invented. And so all of this really changed the depth of information that you could get out and you could find out about lipids. So it's wonderful, but it's huge, it's hugely complicated now. You only got to look at lipid maps to find out how complicated analysis of lipids and how many different lipids there are.
Yeah.
And of course, but it wasn't like that when we started. So I think that's really a good thing. In terms of the redox field, I think the key thing is what I was sort of alluding to early on. When we started, we thought that oxidation was bad and antioxidants were good. Then over time, we realized, not me specifically, but the field realized, that actually there's a correct balance. If you shift the balance a little bit, that can actually be beneficial. So, for example, if you have a low level of oxidation, then that switches on signaling pathways. I mean, it can be through lipoxidation, but oxidants can directly modify cysteines on proteins, and that controls many different signaling pathways like MAP kinases by inhibiting protein tyrosine phosphatases. And then once we'd understood that, people suddenly realized that oxidation is not particularly bad, it's only excess oxidation. And so now we have a much better understanding that redox is actually important in signaling and controls many physiological processes, has an important hormetic effect like hormesis. So a little bit of what's bad for you actually does you good. So a bit of oxidation then upregulates antioxidant pathways. And if you prevent that, people actually cells can't cope as well. And that translates through to actual really physiological things in humans. For example, athletes take antioxidant supplements. Sometimes that actually inhibits their muscle development and the ability of their muscles to cope with oxidative stress during exercise, which was quite a controversial finding at the time.
And you don't have to watch too many adverts on TV to find something about the benefits of this substance, which is an antioxidant, and whether it
Yeah.
is or not is a different question. But
Yes, absolutely.
it's far more nuanced anyway than
Exactly.
the 30-second advert trying to sell you whatever would have you believe.
Indeed.
You were very involved in EpilipidNet and their interest groups. And you've led the Horizon 2020 Master Plan Network, and you do a lot of postgrad and early career researcher training. What would you say the challenges and indeed the joys of doing that training are?
Well, the joys are always imparting knowledge and seeing young early career researchers and young people learn new things and develop and go on to use the knowledge that they've gained in their research. The challenges depend on what the network is. For example, with Master Plan, we had 10 different sites across Europe in 6 different countries. The researchers were registered as PhD students somewhere, but the challenges were really trying to make the different systems fit together. And the regulations for PhD students and training in one country fit in another country, especially because the UK has always been a bit different in its approaches in terms of fee structure and what's required in a PhD. I think in the UK, we see it a little bit more as a training exercise, and it's not so critical for people to have published papers. So trying to bridge the gaps or the differences between different countries was challenging.
Yes, I know there's some European countries you have to have several first author papers in order to get a PhD. And
Yes.
that isn't the case here, definitely, because otherwise
No.
I would have never got mine. But
No, me neither, actually. But there we go. We do try and encourage people now. I think we work much harder now than we used to make sure that early career researchers do get a publication of some kind, whatever it is. So, I mean, the other thing that's challenging, because I've worked quite a lot with mass spectrometry, that's really a very complex area. So you've got to do a lot of very in-depth training to understand the principles of mass spectrometry. And maybe at the basic level, it's relatively simple, but as soon as you actually get into how the instrument works and why these 15 different instruments, can measure different things and how they work, then it's quite a challenging area. And one of the things I found was it was sometimes difficult to persuade people whose projects weren't primarily about mass spectrometry that this was actually a really valuable technique and that they should try and understand it. But I guess that's true with any technique that you're trying to teach people. Unless it's immediately relevant to them, they don't always want to focus on it.
And it's important to know enough about the technique to know its limitations and know how to interpret the data and how
Yes.
not to interpret the data.
Yes, that's always a good point. What does the data really tell you? And especially in lipidomics, have you interpreted it correctly? Have you identified the lipid correctly? To what level have you identified the lipid? I think that's an area of a lot of discussion.
It's a vital area. I think there's I see an awful lot of over-analysis in the literature.
Yes.
And I don't know what we can do about it, but
Education.
Yeah.
It all comes down to educating, and I do have 2 new PhD students starting, and I'm planning some training with them next week on mass spectrometry. But once we get to lipids, it's going to be trying to make them understand what we can actually say from the data that we've got, and try not to overinterpret it. At least, if there are limitations, to say what the limitations are and how that affects our understanding.
And within EpiLipidNet, you developed some outreach materials and games on lipids, didn't you? Can you tell me something about those?
Oh yes, that was really fun actually. I think that was probably one of the most fun networking experiences I've had. And we actually started with the idea that we wanted to explain to people, to the public and to children, what lipids were, and that they weren't all bad, because lipids often get a very bad press, high cholesterol, fat is bad for you. And then we realized that actually there wasn't so much interest in producing flyers or posters, and it would be good to have something interactive. The European Commission organizes quite a lot of European researchers' nights across Europe, and it's really good to have interactive things to do with people who come to visit them. So we started on this concept of games, and in the end, we have 3 games that came out of EpiLipidNet. One was called Lipodrome. And that's it's a bit like Monopoly, where you can go round and round in circles. But it has a start and a finish line.
Mm.
And as you go round, if you land on a blue square, then you pick up a card. And the card says things like, oh, you ate 3 hamburgers for breakfast. That's really not good for you because it's animal fat, etc. Go back 2 places. Or you had beneficial lipids in algae. Different algae that are included in the diet. This is very good for your brain and contains gangliosides. Go forward 2 spaces. So that was very nice. And we've had a lot of interest in Lipodrome. People have used it at the start of different European projects that they're running. People have used it in universities. People have taken it to schools. And then we had a couple of other games as well. One is Know Your Fats, which is quite simple. It's just really a memory game, but it's all about fats. No, fats don't mix with water. Match 2 cards with these symbols. But it's a very simple game that anyone from, young child to elderly adult can play. And the last game I wasn't responsible for. It was the brainchild of someone else, Andre Kuda, and his student Davila. This is called Burn Your Fat, which is far more complicated and is actually about oxidation of lipids in mitochondria, going through all the cycles of NADH to break down the fatty acyl chains. And that is then really targeted almost at university students or perhaps very high level high school children. A nice suite of games that can be used for a variety of different outreach purposes for children to adults.
Oh, these are available, of course, from the Lipid Maps website, but I must admit I've not yet had chance to have a play with them, but they do sound a lot of fun. Earlier, you mentioned you had 2 students, PhD students, starting in the lab. What advice would you give to young scientists starting out in your field in redox biology and lipidomics now? And especially, how do they find their research niche?
Oh, well, I think for PhD students, their research niche is often, that's given to them at the start, and
Yeah.
they develop it throughout their 3 or 4 years. I think for lipidomics or lipid analysis, what I would say is it's a really challenging area. Don't underestimate it. Don't get disheartened either, because it will take you a long time and a lot of work and a lot of reading and practical work to really understand the area and the breadth of the area. I would say lipidomics in the real sense of omics is almost impossible, because you've got to do different experiments. But no, lipids are so diverse. No, they're not like proteins. Proteins are all similar. They're all made up of amino acids with peptide bonds, apart from a few post-translational modifications, etc. But lipids have really diverse structures, and so you need different extraction methods. One extraction method may not be so good for example, sterols or oxysterols compared to fatty acids or phospholipids. So that's really important. And then the type of instrument and the way you set the instrument up will also lead to differences in what you may see, whether you see all the lipids. And actually, you can never see all the lipids at once. You need lots of different experiments really to get a good feeling for what's there. So I have to say, lipidomics is not my favorite thing, really, because it's just so complicated. You almost feel that it's a misnomer. It can't quite be done.
Lipidomics is a completely different ballgame.
Definitely. And once you introduce oxidized lipids and oxlipidomics or redox lipidomics, then you've expanded the complexity enormously.
Yeah, and becomes hugely complex.
The other
Go
thing
on.
I would say for young people is build on your strengths. So when as people move from a PhD to a postdoc and then look for another postdoc or a permanent position, it's good to build on your strengths and to have something that you're comfortable with to carry on doing. But it is also good to diverge a bit and learn new skills and not just to stay doing the same thing all the time. But it is important to have a good track record in what you're doing when you come to applying for different jobs and positions and so on. Then the other thing that I think that is really important is join relevant societies. So in the redox area, we've got the Society for Free Radical Research in Europe, and there are societies all around the world, all under the umbrella organization of SFRR International. If you go to the meetings for the society, and you'll learn a lot about redox biology and the latest thinking. And the same is true for lipids, lipid analysis or mass spectrometry. There we have a British Mass Spectrometry Society, and there are European mass spectrometry societies and American ones. And then we also have the International Lipidomics Society. So I think it's really important to engage with the societies in the field, in the area that you work in, and actually maybe not just one, but a couple of them if you're in a multidisciplinary or cross-disciplinary area. And through that, you'll also meet people who might be your future employers and your colleagues in future years. And building a network is so important for young researchers.
Thanks, Corinne. That's been really interesting. A brief guide into redox biology and redox lipidomics or redox lipid studies. Thanks a lot for your time, and thanks for joining us, and thanks everyone for joining us in the Lipid Maps podcast, and we'll see you again in the next podcast.
Thank you. Goodbye.